There is no blue sky above the first great living world. No forests breathe into the air, no animals stir beneath the surface, and no familiar scent of rain moves across the stone. The young earth turns beneath a dimmer sun, wrapped in an atmosphere that would be instantly fatal to almost every complex organism alive today. Nitrogen is abundant. Carbon dioxide is far more important than it is now. Methane rises from microbial metabolisms and volcanic sources. Water vapor gathers, falls, evaporates, and returns again. Oxygen exists only in traces, quickly consumed whenever it appears. The planet is alive, but it is alive in a language modern lungs cannot speak. More than 3 billion years ago, the seas already contain communities of microorganisms that have survived for immense spans of time. Their world is not empty or primitive in the dismissive sense. It is chemically active, ecologically varied, and crowded with invisible competition. Some microbes draw energy from hydrogen, others use iron, sulfur compounds, or carbon dioxide. Methanogens produce methane as part of their metabolism, feeding a greenhouse that helps keep surface water liquid beneath a sun substantially fainter than the one shining now. Organisms exchange chemicals across gradients in mud, shallow water, hydrothermal systems, and mineral-rich coastlines. Life has learned to inhabit a planet without breathable air, because breathable air has never been part of its history. The continents are smaller, darker, and more barren than anything a human traveller would recognize. There is no soil built by roots, no moss softening exposed rock, and no vegetation holding slopes in place. Rain strikes naked mineral surfaces and carries dissolved material toward the oceans. Volcanoes release gases from Earth's interior, including hydrogen, carbon monoxide, sulfur species, and other reduced compounds eager to react with oxygen. Iron washes into the seas and remains dissolved because the deep water contains almost no free oxygen to turn it into rust. In many places, the ocean may appear greenish beneath microbial blooms or dark with suspended particles, though the evidence remains incomplete, and no single reconstruction can recover its exact colours. What survives in the rocks instead reveals a world maintained by chemical balances very different from those of the present. For anaerobic organisms, oxygen is not a promise of future complexity. It is a dangerous oxidant. It attacks cellular components, alters proteins, damages genetic material, and disrupts metabolisms that evolved in its absence. Modern people associate oxygen with life because their bodies depend upon it, but this relationship is historically late. For much of Earth's biological past, oxygen is closer to contamination. The organisms dominating the planet do not merely tolerate a world without it, they require that world. Their enzymes, membranes, and energy pathways are fitted to conditions in which reactive oxygen remains rare and temporary. Even so, the ancient atmosphere is not perfectly uniform. Lightning splits molecules, ultraviolet radiation drives reactions high above the surface, water can be broken apart, and a small portion of hydrogen escapes to space, leaving behind oxidizing potential. Local oxygen may also form through biological activity before it ever becomes a stable atmospheric presence, yet nearly every molecule enters a landscape filled with sinks waiting to consume it. Reduced volcanic gases react with it, dissolved iron binds it, sulfur compounds take it up. Organic matter is oxidized. Any oxygen that reaches seawater can be used by microbes living close to its source. The atmosphere remains almost entirely anoxic, not because oxygen is impossible, but because the planet is extraordinarily efficient at removing it. This distinction matters. The later transformation of Earth does not begin with the silent planet suddenly discovering oxygen. It begins with oxygen appearing in places where it cannot yet endure. Long before the atmosphere changes globally, small zones may flicker with a chemistry that belongs to the future. Around microbial mats in sunlit shallows, oxygen can rise during periods of active photosynthesis and vanish again when darkness returns or when surrounding materials absorb it. These temporary pockets are sometimes described as oxygen oases. They do not make the world breathable. They make the world uneven. The old biosphere thrives within that unevenness. At the surface, sunlight powers microbial communities layered by their tolerance for light and chemistry. Below them, other organisms consume fermentation products, sulfur compounds, hydrogen, or methane. Along the seafloor, hydrothermal vents supply reduced chemicals from the crust. In sediments, communities occupy narrow bands only millimeters or centimeters apart, each shaped by what the layer above produces and what the layer below can use. Even in a world of single-celled life, existence is organized around boundaries. A tiny shift in chemistry can create a new habitat or erase an old one. Some of the clearest monuments to these communities are stromatolites. Laminated structures built as microbial mats trap sediment and influence mineral precipitation. They are not the remains of single creatures, but the accumulated architecture of communities. Layer by layer, they preserve evidence that life was already reshaping its surroundings. Yet stromatolites alone cannot reveal which organisms lived in every layer or exactly what metabolisms they used. Scholars still debate how early oxygen-producing photosynthesis evolved and how widely it spread before the major atmospheric transition. The fossils provide form, chemistry and molecular evolution provide other pieces. None offers a complete picture by itself. The planet's energy economy is therefore both stable and vulnerable. Methane helps warm the climate. Carbon dioxide also contributes to greenhouse conditions. The oceans hold vast reservoirs of dissolved iron and reduced chemicals. Microbial ecosystems recycle nutrients under low oxygen conditions. For hundreds of millions of years, these relationships support a functioning biosphere. Nothing within that world suggests that oxygen must eventually dominate. Evolution has no plan and no obligation to prepare one age for the next. Then a metabolism appears, or perhaps slowly spreads. That changes the meaning of sunlight. Certain microbes learn to use water as an electron source in photosynthesis, releasing molecular oxygen as a byproduct. Water is abundant, and the innovation is powerful, but its consequence is not immediate. The oxygen disappears into iron, sulfur, rock, gases, and living cells almost as quickly as it is made. The ancient world absorbs the disturbance and continues. For a time, that may seem like the end of the story. A new waste product enters the environment. The environment neutralizes it. Yet every sink has a rate. Every chemical reservoir has limits. And every biological innovation can alter the conditions that once contained it. Its quiet waste will become a geological force. Beneath the unbreathable sky, the first tension begins to build between a biosphere adapted to oxygen's absence and organisms capable of producing it without intending any planetary revolution. The world has not yet turned against anaerobic life, but the machinery of that reversal is already alive in the shallows. The machinery begins with a problem older than forests, animals, or even oxygen itself. How can a living cell capture enough energy from sunlight to survive? Early photosynthesis does not necessarily release oxygen. Some microbes use hydrogen sulfide, ferrous iron, or other reduced compounds as electron donors, turning light into chemical energy without splitting water. These metabolisms work, but their raw materials are limited and unevenly distributed. Water is different. It covers much of the planet. To draw electrons from it requires an elaborate molecular system, but once that system evolves, sunlight can be coupled to one of Earth's largest reservoirs. The price of that achievement is oxygen. The organisms associated with this innovation are cyanobacteria, though their earliest history remains difficult to reconstruct. Their cells are microscopic, their fossils ambiguous, and the genetic record has been altered by billions of years of evolution. What survives in the geological record suggests that oxygenic photosynthesis emerged well before oxygen accumulated permanently in the atmosphere, perhaps hundreds of millions of years earlier. Scholars still debate the timing, and no single fossil or molecular clock settles the question. Yet the broad pattern is increasingly difficult to avoid. Biological oxygen production precedes the Great Oxidation Event by a long interval. Cyanobacteria do not set out to transform the planet. They are solving a local metabolic problem inside their membranes. Linked photosystems absorb light energy and use it to pull electrons from water. Carbon dioxide is converted into organic matter, building cells and storing energy. Oxygen is expelled as waste. It diffuses into surrounding water, reacts with dissolved iron, oxidizes reduced sulfur compounds, and is consumed by nearby organisms capable of using it. In sunlit microbial mats, oxygen concentrations may rise during the day and collapse after sunset as respiration continues without photosynthetic production. This makes the ancient shoreline a place of extraordinary contrast. A microbial film only a few millimeters thick and contains several chemical worlds. At the top, cyanobacteria receive sunlight and release oxygen. Beneath them, oxygen falls away sharply. Deeper layers support sulfur-cycling microbes, fermenters, and methanogens that would be harmed by prolonged exposure to the gas produced above. The mat survives because chemistry is stratified. Oxygen does not immediately invade everything. It forms a narrow frontier, bright in energy but dangerous in consequence. Before we go deeper, take a moment to like the video and subscribe. If this is the kind of history and mystery you enjoy, the surprising possibility is that some organisms learn to exploit oxygen long before the atmosphere contains much of it. Molecular clock research on hemicopper oxygen reductases, enzymes involved in oxygen-based respiration, suggests that certain oxygen-using systems may have diversified during the Archean, perhaps between about 3.2 and 2.8 billion years ago. The earliest oxygen may be consumed almost where it is produced. Microbes living beside cyanobacteria gain access to a powerful electron acceptor and turn the new gas into metabolic energy. In doing so, they help prevent it from escaping into the wider ocean and atmosphere. Oxygen therefore begins as both poison and opportunity. To strict anaerobes, it threatens cellular machinery. To organisms with protective enzymes and suitable respiratory pathways, it offers an energetic advantage. Aerobic respiration can extract far more usable energy from organic matter than many anaerobic pathways. Around each oxygen-producing community, selection favors defenses against reactive oxygen, mechanisms for repairing molecular damage, and eventually metabolisms that make use of the danger itself. Still, local production is not global transformation. The oceans contain enormous quantities of dissolved ferrous iron. When oxygen encounters that iron, the iron is oxidized and becomes less soluble. It precipitates as iron-bearing minerals that settle toward the seafloor. Repeated over vast spans of time, this process contributes to the layered deposits later known as banded iron formations. Even so, they are immense evidence that oxygen was being generated and consumed in marine environments long before the air became familiar. Volcanic gases provide another sink. Earth's interior releases reduced compounds that react readily with oxygen. Hydrothermal fluids deliver iron and sulfur to the sea. Organic matter produced by photosynthesis is often decomposed, consuming oxygen or returning carbon to forms that do not allow atmospheric oxygen to accumulate. For oxygen to build up, it is not enough for cyanobacteria to produce it. The rate of production must exceed the combined demand of geology, chemistry, and biology. This is why the rise of oxygen is less like filling an empty vessel, and more like trying to flood a landscape filled with drains. Some drains are vast reservoirs of reduced material, others are continuous flows from volcanoes, weathering reactions, and microbial respiration. As production increases, the biosphere changes, nutrient cycles shift, and new oxygen-consuming metabolisms emerge. A model proposed in 2025 even suggests that an early origin of oxygenic photosynthesis could delay atmospheric oxygenation by expanding a biosphere that recycles nutrients and consumes oxygen efficiently. The invention that makes the Great Oxidation possible may also help postpone it. Phosphorus becomes especially important. It is required for cells, genetic material, and energy transfer. Where bioavailable phosphorus increases, photosynthetic productivity can rise. Recent work on Archean oxygen oases suggests that pulses of phosphorus recycling may have fueled temporary blooms of oxygen production before the global transition. These episodes leave scattered chemical hints, sometimes called oxygen whiffs, in rocks older than the conventional beginning of the Great Oxidation Event. Their interpretation remains contested in detail, but they fit a world in which oxygen appears repeatedly without yet taking control. The cyanobacteria continue to spread through suitable waters, constrained by nutrients, light, and temperature. The cumulative effect is geological. Trillions of cells split water, fix carbon, and discard oxygen. Most molecules vanish into sinks. A small fraction alters minerals. Another fraction supports oxygen-using organisms. Some organic carbon is buried before it can be fully oxidized, leaving the released oxygen chemically unmatched. That burial is crucial. Photosynthesis produces oxygen and organic matter together. If the organic matter is later consumed through reactions that use oxygen, the apparent gain is cancelled. When organic carbon is buried in sediment and separated from the surface system, some oxygen can remain behind. The planet's redox balance shifts by increments too small for any organism to perceive. Over millions of years, those increments matter. The old anaerobic world is not conquered by a sudden invasion. It is undermined by repetition, day after day. Microbes use water and light in a reaction whose waste the planet can still absorb. Nutrients arrive and disappear. Iron precipitates. Gases rise from volcanoes, and oxygen blooms around microbial communities before fading again. But the chemical debt is changing. The sinks are being altered, the sources are becoming more persistent, and life has discovered a metabolism whose raw material is nearly inexhaustible. The revolution remains invisible from space. At the scale of a cell, however, it is already underway. The first oxygen does not rise triumphantly into the sky. It vanishes. Every molecule enters a planet loaded with substances eager to consume it. allowing oxygenic photosynthesis to exist for immense spans without producing an oxygen-rich atmosphere. The ancient Earth behaves like a vast chemical sponge. Its oceans, crust, volcanic gases, sediments, and living communities absorb the new gas faster than it can accumulate. What cyanobacteria release into sunlit water is often trapped before it travels more than a short distance. The largest visible sink lies dissolved in the sea. Archean oceans contain abundant ferrous iron, carried from hydrothermal systems and weathered rock under conditions where free oxygen is scarce. In its reduced form, this iron remains soluble and can spread through seawater. When oxygen reaches it, the reaction changes the iron into less soluble oxidized forms. Tiny mineral particles appear, sink, and accumulate on the seafloor. Over time, layers rich in iron alternate with layers containing silica and other material, producing the striking banded iron formations that now survive in ancient crust. These deposits are sometimes imagined as the rust of an entire ocean, but the real process is more complicated. Iron precipitation may have occurred in pulses, in particular basins, and under changing biological and chemical conditions. Scholars still debate how much each mechanism contributed in particular formations, yet the broad significance remains powerful. The ocean contained enormous reservoirs of reduced iron, and oxygen entering those waters could be consumed on a planetary scale. A shoreline observer, had one existed, would not have watched the sea turn red in a single dramatic season. The transformation unfolds across intervals longer than species endure. In shallow water, near photosynthetic mats, oxygen rises during daylight and reacts with iron, arriving from deeper layers or nearby hydrothermal sources. Minerals form, settle, and are buried. Currents mix water masses with different chemistries. Storms disturb sediments and expose fresh reduced material. At night, respiration and chemical reactions draw oxygen back down. The same place can shift repeatedly between oxygenated and anoxic conditions without ever resembling a modern coastal sea. The crust itself presents another vast demand. Fresh basalt, volcanic ash, and exposed minerals contain reduced iron and other compounds capable of reacting with oxygen. Wherever new rock is erupted or uncovered, it offers another set of chemical bonds waiting to be altered. Weathering redistributes elements, changes ocean chemistry, and feeds the balance between oxygen sources and sinks. Volcanoes make that balance even harder to overturn. Their gases include hydrogen, carbon monoxide, hydrogen sulfide, and other reduced species that react readily with oxygen. The exact composition of ancient volcanic emissions remains uncertain because it depends on mantle chemistry, pressure, tectonic setting, and how gases interact with magma and water before reaching the atmosphere. Even modest changes in volcanic gas composition could alter how much oxygen survives. A more reducing flux consumes oxygen aggressively. A less reducing flux allows biological production to gain ground. This creates a strange contest between Earth's interior and its surface biosphere. Cyanobacteria use sunlight to split water and release oxygen. Volcanoes and hydrothermal systems deliver reduced material from below. The atmosphere and ocean sit between them, receiving products from both. For long periods, the rates nearly balance. Oxygen is produced, but its presence is local, temporary, or recorded only through altered minerals. The sky remains largely anoxic because the planet's deep interior continues to supply a chemical counterweight. Organic matter is another drain. Photosynthesis creates carbon-rich biomass at the same time that it releases oxygen. If that biomass is later decomposed through oxygen-consuming respiration, the oxygen gain disappears, even without abundant atmospheric oxygen. Microbial communities can recycle organic matter through complex chains of fermentation, sulfur reduction, methanogenesis, and local aerobic respiration. Only when some organic carbon escapes decomposition and is buried in sediment does the oxygen associated with its production remain available to the surface environment. Burial is therefore a quiet form of separation. A fraction of dead microbial material settles into mud, becomes shielded from oxidants, and is eventually incorporated into rock. The carbon is removed from short-term circulation. It may later return through metamorphism, erosion, or volcanism, but for a time its burial leaves behind an oxidizing effect. The efficiency of this process depends on sedimentation, nutrient supply, ocean chemistry, and the activity of organisms consuming organic matter. No single explanation fully accounts for how these variables changed enough to permit persistent oxygenation. Sulfur adds still another layer. Reduced sulfur compounds consume oxygen, while the oxidation of sulfide minerals on land produces sulfate that rivers carry to the sea. Before significant atmospheric oxygen, oxidative weathering is limited. After oxygen begins to rise, sulfur cycling changes in ways preserved by isotope patterns in ancient rocks. Those patterns become some of the most important clues to atmospheric chemistry, but they do not translate directly into a simple oxygen percentage. They reveal thresholds, pathways, and departures from earlier conditions rather than a precise reading from an ancient instrument. The same caution applies to every mineral archive. A red stain in rock records oxidation, but not necessarily a globally oxygen-rich atmosphere. An iron formation records changing ocean chemistry, but not a uniform ocean. A sulfur isotope signal may show that oxygen crossed a critical threshold in the air, yet local environments can still remain deeply anoxic. Earth's surface is a mosaic, and geological evidence samples only fragments of it. For hundreds of millions of years, oxygen production may increase without overwhelming this network of sinks. Iron continues to precipitate. Fresh volcanic material continues to react. Reduced gases continue to rise. Organic matter is produced and largely recycled. Oxygen-using microbes take advantage of local concentrations and consume part of the supply. Each process helps explain why biological innovation precedes atmospheric revolution by such a long interval. But the sinks are not timeless. Dissolved iron reservoirs can be depleted or reorganized. Volcanic gas fluxes can change as the mantle and crust evolve. Continents grow, weathering patterns shift. Nutrient delivery alters productivity, and burial removes reduced carbon from circulation. The balance does not need to change dramatically in a single moment. It only needs to tilt persistently enough so that a little more oxygen survives than before. The stone record is the memory of that long resistance. Layer after layer captures oxygen being spent rather than saved. The early atmosphere remains hostile to modern lungs because oceans and rocks absorb the waste of photosynthesis with extraordinary success. Its success is also the measure of coming change, yet in doing so, they are slowly transformed. Iron is removed from solution. Minerals are oxidized. Sediments store buried carbon. The very sinks that delay oxygen's rise are altered by the oxygen they consume. Eventually, the planet's capacity to hide the gas begins to weaken, and chemistry that once erased every trace starts leaving something behind. By the time oxygen begins to leave a more persistent mark, the planet has already spent hundreds of millions of years hiding the evidence. The atmosphere itself does not fossilize. No bubble of Archean air waits intact for scientists to open. Instead, the story survives indirectly, written in minerals, sediments, isotopes, and chemical imbalances that were locked into rock before later heat and pressure could erase them. Reconstructing the rise of oxygen is therefore less like reading a diary than examining a crime scene after most of the room has been rebuilt. Banded iron formations are among the most dramatic witnesses. Their alternating layers of iron-rich minerals and silica record repeated changes in ancient seawater, but they do not provide a simple date for when the atmosphere became oxygenated. Many formed long before the Great Oxidation Event, when oxygen produced in local surface waters reacted with dissolved ferrous iron. Their very abundance shows how effectively oxygen was consumed. Later changes in their distribution suggest that ocean chemistry was being reorganized. Yet deep waters could remain anoxic even as surface environments became more oxidizing. Another clue lies in sulfur. Sulfur has several stable isotopes, and in the absence of significant atmospheric oxygen, ultraviolet light can drive chemical reactions that separate those isotopes in unusual ways. The resulting pattern, known as mass-independent fractionation, can be preserved in sedimentary rocks. For much of the Archean, that signal appears widely. Then, around the broad interval associated with the Great Oxidation Event, it largely disappears. This loss is one of the strongest indicators that atmospheric chemistry crossed an important threshold. The reason involves both oxygen and ozone. When oxygen remains extremely scarce, ultraviolet radiation reaches deeper into the atmosphere and produces sulfur reactions that create distinctive isotope anomalies. As oxygen increases, even to levels far below those of the modern world, ozone begins to form and changes how ultraviolet light penetrates the air. Sulfur compounds are also more readily oxidized and removed from the atmosphere. The unusual isotope signal can no longer be generated or preserved in the same way. Its disappearance does not reveal an exact oxygen concentration, but it shows that the atmosphere has entered a new chemical regime. That transition is not perfectly clean. Some rock sequences preserve evidence suggesting that oxygen rose, fell, and perhaps crossed critical thresholds more than once. Deposits from different basins do not always tell identical stories. Erosion, metamorphism, local chemistry, and gaps in sedimentation complicate the record. Scholars still debate whether certain signals represent global atmospheric change, regional oxygen oases, or later alteration. The evidence remains incomplete because the surviving crust from this age is scattered and heavily reworked. Chromium, molybdenum, uranium, vanadium, thallium, and other elements provide additional tools. Their behavior changes depending on oxygen conditions, weathering, and the chemistry of seawater. When sulfide minerals on land begin to oxidize, rivers carry altered metals and sulfate toward the oceans. When manganese oxides form in oxygenated water, they can remove or redistribute trace elements in ways later preserved in sediment. Each proxy responds to different thresholds and environments, so no single one can reconstruct the entire planet. Every conclusion depends on comparison across distant formations, because a single basin may preserve local chemistry while appearing to speak for the whole planet. This is why recent studies increasingly describe the Great Oxidation not as one sharp step but as a prolonged sequence. One set of rocks may record atmospheric oxygen becoming more persistent. Another may show surface ocean oxygenation spreading later. A third may indicate that deep marine environments remained reducing. In some intervals, atmospheric oxygen may still have been astonishingly low, perhaps far below 1,000th of the modern level, even while oxidative weathering had begun to leave detectable marks. Around 2.3 billion years ago, several lines of evidence begin to converge more clearly. Thallium isotopes suggest that atmospheric and oceanic oxygenation became more strongly coupled. Vanadium isotope work indicates that persistent oxygenated surface waters expanded on a larger scale by roughly 2.32 billion years ago, perhaps reaching dissolved oxygen concentrations of at least several micromoles in some marine settings. These were not modern oceans. Oxygenated shallows could lie above deeper waters that remained entirely anoxic and rich in dissolved iron. On land, the appearance of ancient red beds offers a more visual clue. Their colour comes from oxidized iron minerals coating sediment grains. Red beds around 2.3 billion years old suggest that oxygen was affecting exposed continental surfaces and the products of weathering. The image is striking barren landscapes that had long been grey, black, or greenish in mineral tone begin to preserve red oxidation. Yet even here, caution is necessary. Red rock indicates oxygen in a particular weathering environment, not a fully breathable sky. The geological record therefore reveals a world changing by thresholds. First, oxygen is produced but disappears locally. Then it leaves mineral traces in marine basins. Later, atmospheric chemistry changes enough to erase the old sulfur isotope pattern. Oxidative weathering strengthens on land. Surface waters hold oxygen for longer periods. Deep oceans lag behind. The boundaries between these stages overlap, blur, and vary from place to place. This complexity explains why dating the Great Oxidation depends on definition. If the beginning is placed at the first major loss of mass-independent sulfur fractionation, one date may be chosen. If it is tied to persistent atmospheric oxygen, expanding surface ocean oxygenation, or changes in continental weathering, another date may appear more appropriate. If the end is defined by the stabilization of oxygen after later fluctuations, the interval may extend toward about 2.06 billion years ago. The familiar phrase 2.4 billion years ago remains useful shorthand, but it compresses a transformation lasting hundreds of millions of years. The rocks do not describe a switch being thrown. They describe pressure building, thresholds being crossed, and systems responding at different speeds. Their clues are powerful precisely because they disagree in revealing ways. The atmosphere changes before the entire ocean follows. The surface ocean changes before the deep sea. Continents begin to rust while vast habitats remain chemically ancient. What emerges is not a single moment as planetary awakening, but a prolonged instability in which oxygen repeatedly appears, retreats, and returns. By reading these scattered signatures together, scientists can see the outline of a revolution that no living observer recorded. The planet is still largely alien, still dangerous to modern lungs, and still dominated by microbes. But the old chemical order is losing its permanence. Oxygen has moved beyond fleeting oases. It has begun to alter the sky, the continents, and the upper ocean in ways that can no longer be completely erased. The evidence is fragmentary, yet its message is unmistakable. Earth has crossed into a new era, even if the new era has not yet decided what it will become. The new era does not arrive with a clean horizon. It advances through instability. Oxygen has begun to survive beyond microbial mats, yet every increase alters the system that permits it. More oxygen changes weathering. Weathering changes the delivery of nutrients and metals to the sea. Those changes affect photosynthetic productivity, organic carbon burial, volcanic gas consumption, and the organisms that use or avoid oxygen. Between roughly 2.43 and 2.06 billion years ago, the planet appears to pass through several episodes of oxygenation across widely separated parts of Earth, rather than one smooth ascent. The familiar date of 2.4 billion years ago marks the beginning of a broad interval, not the instant when Earth suddenly acquires an oxygen-rich sky. Some records suggest oxygen rises, falls, and rises again. Climate variability, nutrient supply, continental weathering, volcanic emissions, and biological productivity may all help drive these fluctuations. At first, even a small increase in atmospheric oxygen has disproportionate consequences. The modern atmosphere contains about 21% oxygen, but the ancient transition begins at concentrations many orders of magnitude lower. Tiny amounts can alter sulfur photochemistry, permit traces of ozone to form, and increase oxidative weathering on land. Rivers begin carrying more sulfate and oxidized metals into the oceans. Minerals that once remained stable at the surface break down differently. The chemistry of rain, barren rock surfaces, and coastal sediment starts to change long before any animal could breathe the air. This change may reinforce itself. When oxygen promotes weathering of phosphorus-bearing minerals, more phosphorus can reach the ocean. Phosphorus is a limiting nutrient for many photosynthetic organisms. Greater availability may increase biological productivity, allowing cyanobacteria to fix more carbon and release more oxygen. If a larger fraction of that organic carbon is buried before decomposition, oxygen gains another advantage. Recent reconstructions suggest that marine phosphorus and atmospheric oxygen were closely coupled during the Great Oxidation Interval. Though no single nutrient cycle controls the event, phosphorus itself does not exist in only one useful form. Evidence from ancient sediments indicates that phosphite, a reduced form unusual in modern oxygenated surface waters, may have represented a substantial fraction of dissolved inorganic phosphorus in some environments. Microbes capable of using it would inhabit a nutrient world unlike the one dominating today. This matters because the biosphere changing the atmosphere is chemically unfamiliar. It is not simply a modern ecosystem with fewer species. Its pathways and limitations belong to another planet. As oxygen spreads through the upper ocean, the deep sea remains largely anoxic. Sunlit waters can retain oxygen for longer periods, especially where circulation separates them from reduced deep water. Below, iron-rich or sulfidic conditions continue. The boundary between these layers may rise, sink, or move across continental shelves as climate and circulation change. One basin can become more oxygenated at the surface while remaining hostile to oxygen-dependent life at depth. Another may stay reducing almost throughout. The Great Oxidation is global in consequence but uneven in expression. Around 232 billion years ago, evidence from vanadium isotopes points to the expansion of persistently oxygenated surface ocean environments. Some waters may have held at least about 10 micromoles of dissolved oxygen, enough to establish a stable oxidative presence, while remaining far below concentrations typical of modern well-ventilated seas. This is a decisive development. Oxygen no longer appears only as a brief daytime pulse beside a microbial mat. In some regions, it endures. Yet endurance is not dominance. Studies of sulfide weathering suggest that atmospheric oxygen during portions of the event may still have remained below roughly 5/10,000ths of the modern atmospheric level. Estimates vary widely, sometimes by several orders of magnitude, because proxies respond to different processes and thresholds. Scientists can identify chemical transitions while remaining uncertain about the exact amount of oxygen in the air. The word oxidation can sound more complete than the reality. The atmosphere is changing, but it is still almost entirely unbreathable. The sources of oxygen also face constant resistance. Reduced volcanic gases continue to consume it, organic matter is recycled, iron and sulfur reservoirs remain active. Oxygen-using microbes expand wherever the gas becomes available and turn part of the supply into respiration. In this sense, biological success creates its own break. The more oxygenated niches appear, the more organisms evolve to exploit them, increasing oxygen demand. A planetary rise requires production and burial to stay ahead of a growing community of consumers. Climate adds another source of instability. Cooler conditions can alter weathering, ocean circulation, nutrient delivery, and biological productivity. Glaciation can suppress some processes while exposing fresh rock during retreat. Warmer intervals can accelerate weathering and nutrient fluxes, but also strengthen oxygen consumption through greater productivity and decomposition. Recent modelling suggests that climate variability may have generated multiple oxygenation episodes across the event. The atmosphere moves through feedbacks that can push it forward, stall it, or force temporary retreat. Ozone begins to matter even when oxygen remains scarce, formed from molecular oxygen under ultraviolet radiation. Ozone absorbs some damaging wavelengths and changes atmospheric photochemistry. Its emergence is not yet a shield comparable to the modern ozone layer, but it alters which reactions occur high above the surface. Sulfur compounds behave differently. Methane chemistry changes. The lifetime of gases shifts. A biological waste product begins controlling the light environment and chemical architecture of the sky. By about 2.3 billion years ago, the coupling between atmospheric and marine oxygenation becomes more apparent in several geochemical records. Oxidative weathering on land strengthens, oxygenated surface waters expand, and the old sulfur isotope signature fades. But the process remains reversible enough to preserve later declines. The Great Oxidation is great not because oxygen instantly reaches modern abundance, but because Earth crosses from a world where free oxygen cannot persist globally to one where it becomes a recurring and durable feature of the surface system. That threshold transforms the direction of planetary history. Before it, oxygen is a local disturbance repeatedly erased by rock, water, gas, and metabolism. During it, the same sinks still operate, but they can no longer restore the old equilibrium everywhere. The atmosphere begins carrying oxygen from one region to another. Continents record its touch. Surface oceans retain it after daylight ends. Each pulse makes the next pulse chemically easier in some places and biologically more disruptive in others. The old world has not vanished. Anaerobic metabolisms remain widespread, and most of the ocean still belongs to them. Yet permanence has been lost. A gas once confined to microscopic frontiers now participates in global cycles. Its concentration may be low, uncertain, and unstable, but its reach is expanding. What began as metabolism has become climate, weathering, ocean chemistry, and planetary memory. Earth is not yet an oxygen world in the modern sense. It is something more precarious, a world caught in the act of becoming one. The crisis begins at boundaries too small to see. In a microbial mat, oxygen produced near the sunlit surface diffuses downward into layers inhabited by organisms whose chemistry evolved in its absence. To those cells, the gas is reactive. Oxygen can generate radicals that damage proteins, membranes, and genetic material. Enzymes built for an anoxic world may be disabled. A habitat stable for millions of years can become dangerous because a neighboring community has learned to split water. This does not mean every anaerobe dies wherever oxygen appears. Many strict anaerobes retreat into deeper sediments, stagnant waters, hydrothermal systems, or the interiors of microbial communities where oxygen is consumed before reaching them. Others evolve defenses. Protective enzymes neutralize reactive oxygen, though their early history remains uncertain. Some organisms tolerate brief exposure without using oxygen. Others learn to exploit it. The biosphere reorganizes along gradients of exposure, protection, and metabolic opportunity. The transformation is therefore ecological before it is global. An oxygenated surface layer forms over an anoxic sea. River mouths, shallow shelves, tidal flats, and microbial mats become zones where chemistry changes over hours, seasons, or geological intervals. A population adapted to reduced water may find its range compressed as oxygen persists longer. Another may expand because oxygen removes toxic sulfide from its surroundings. For strict anaerobes, however, the direction of change is unmistakable. Their habitats become fragmented. Places once connected through a continuous low-oxygen ocean are separated by oxidized surface waters or sediments. The most favorable zones move downward, offshore, or into restricted basins. Ancient metabolisms survive, but their geography narrows. The world that had been broadly compatible with anaerobic life begins to reserve its safest spaces for darkness, depth, burial, and isolation. This is why calling the event a poisoning captures part of the truth, but not all of it. Oxygen is toxic to many organisms, yet toxicity depends on concentration, duration, and cellular defenses. A trace amount can damage one species while powering the metabolism of another. Oxygen also changes the environment indirectly. It oxidizes dissolved iron, alters sulfur compounds, suppresses methane production in exposed zones, and changes which nutrients remain available. Organisms may suffer not from direct exposure but because the food web and geochemical cycles supporting them are being rearranged. The fossil record cannot provide a census of these losses. Most organisms are microscopic, soft-bodied, and unlikely to fossilize. Their ecological collapse does not leave piles of skeletons. Instead, scientists infer biological change from isotope patterns, mineral deposits, biomarkers, and shifts in productivity. The evidence remains incomplete, and no single explanation fully accounts for the scale or timing of biospheric disruption. It is unsafe to claim that oxygen nearly exterminated all life. What survives in the record suggests something subtler. The dominant organization of life was forced to change. Some anaerobic communities may have declined sharply as oxygenated habitats expanded. Others may have persisted almost untouched in deep sediments and subsurface environments. Methanogens, important producers of atmospheric methane, likely faced pressure where oxygen penetrated. Because methane is both a metabolic product and a powerful greenhouse gas, changes in microbial habitat could influence climate far beyond the cells directly affected. The contraction of one ecological guild can become an atmospheric event. At the same time, oxygen opens a new energetic frontier. Organisms capable of aerobic respiration can extract much more usable energy from organic molecules than many anaerobic pathways allow. That advantage does not instantly produce animals or complex cells, but it changes the possible economics of life. More energy can support faster growth, active transport, repair systems, and biochemical complexity. The gas threatening the old biosphere becomes a resource for organisms able to control it. The first steps toward that control may predate the Great Oxidation by hundreds of millions of years. If oxygen-reducing enzymes diversified in Archean environments, then microbes had already been experimenting with oxygen long before the atmosphere retained it. They lived near oxygen oases, perhaps using tiny concentrations produced during daylight. Their existence makes the crisis more complicated. Oxygen does not arrive in a world entirely unprepared. Pockets of adaptation already exist, ready to expand when the gas becomes persistent. Selection favors several strategies. Some cells remain hidden in anoxic refuges, some build chemical defenses, some migrate across microscopic gradients, some consume oxygen before it reaches sensitive neighbors, some use it as the final electron acceptor in respiration. Communities may even protect their anaerobic members collectively, with surface organisms removing oxygen and preserving deeper layers. Survival can be an emergent property of a layered ecosystem. Yet protection has limits. As oxygen persists in surface waters and weathering products carry oxidized compounds into the sea, the frontier moves. The daily oxygen pulse becomes seasonal, then regional, then geologically durable in some settings. A refuge that once began a millimeter below a microbial mat may retreat deeper. A basin once rich in dissolved iron may develop oxygenated margins. Sediments once chemically uniform become divided into sharp redox zones. Life is compelled to follow those boundaries or perish locally. There is also a temporal mismatch. Evolution can respond, but atmospheric and geochemical feedbacks may change conditions faster than some populations can adapt. A lineage optimized for one narrow redox state may not possess the biochemical tools to survive another. Local extinctions almost certainly occur, though their number cannot be measured. Genetic innovations spread where they confer protection or access to new energy. Horizontal gene transfer may help distribute useful metabolic machinery among microbes, allowing adaptation to cross lineage boundaries. The result is not a silent emptying of the planet, it is a turnover in power. Anaerobic life remains abundant, but it no longer defines every surface environment. Oxygen-tolerant and oxygen-using organisms gain territory. Ecological interfaces multiply. New metabolisms arise around nitrate, sulfate, iron, and oxygen cycling. The biosphere becomes more chemically partitioned, more energetically varied, and more dependent on interactions between incompatible forms of life. No observer witnesses the transition as catastrophe. There is no single generation in which the old world ends. The poisoning unfolds across vast intervals, through local crises repeated in oceans and sediments. A microbial community disappears from one shoreline, survives in another basin, and persists deep underground beyond the reach of atmospheric change. The paradox is that life both suffers from oxygen and learns to build a larger future with it. By the time oxygen becomes a recurring feature of the surface environment, the ancient biosphere has not been destroyed. It has been displaced, divided, defended, and transformed. The organisms that once occupied nearly every habitable chemical niche are pushed toward refugees, while new lineages colonize the expanding oxidative world. Earth's atmosphere has turned against anaerobic life, not as an executioner acting in a moment, but as a changing landscape that slowly withdraws the conditions on which an older order depended. The planet's transformation becomes visible not because the sky suddenly turns blue, but because stone begins to remember oxygen across exposed land. Iron-bearing minerals react with the new oxidant and turn red, orange, and brown. In the oceans, dissolved ferrous iron is converted into insoluble minerals and drawn out of the water column. Sediments change color. Weathering changes pace. Rivers carry different chemical loads toward the sea. Earth is not merely gaining a gas, it is repainting its surface through reactions that had once been rare. Before widespread oxygenation, much of the iron delivered to the oceans can remain dissolved in its reduced form. Hydrothermal systems supply it from the crust, and anoxic deep waters allow it to travel great distances. Oxygen released as the upper ocean meets this iron and strips it from solution. Particles form, settle, and accumulate. The process contributes to immense iron-rich deposits that later become economically important to a species not yet imaginable. Modern cities and machines will eventually depend on ores created partly by microbial waste billions of years earlier. The image of ancient seas turning rusty is useful, but it can also mislead. Oxidation does not happen uniformly across every basin. Some waters remain iron-rich, others become sulfidic, and shallow oxygenated zones may sit above deeply anoxic layers. Currents move chemical boundaries. Seasonal productivity shifts them. Local nutrient supplies determine how much oxygen cyanobacteria can produce. What survives in the record suggests a patchwork planet, with redox fronts advancing and retreating rather than one ocean changing colour at once. On land, the emergence of red beds around 2.3 billion years ago offers a striking sign that atmospheric oxygen is affecting continental surfaces. Iron within sand grains and sediments oxidizes, forming hematite and related minerals that stain the rock. These red layers do not prove that oxygen levels approached anything breathable. They show that oxygen had become persistent enough locally to alter exposed sediment before burial. A barren river plain under a dim sun could now carry a colour associated with modern deserts. Weathering also changes what the continents export. As sulfide minerals oxidize, sulfate enters rivers and eventually reaches the sea. Metals bound in rocks become more mobile or are trapped in new mineral forms. Phosphorus can be released, but its fate depends on iron chemistry, sedimentation, and biological uptake. Oxygen therefore creates competing effects. It can increase the delivery of nutrients from land, encouraging photosynthesis, while also causing phosphorus to bind to oxidized iron minerals and disappear into sediment. The same process can feed productivity in one setting and starve it in another. This tension helps explain why oxygen does not rise without limit. More oxygen strengthens oxidative weathering. Weathering supplies nutrients that can stimulate cyanobacteria and increase oxygen production. Yet stronger oxidation also changes marine mineral surfaces and can remove phosphorus from circulation. If productivity falls, oxygen production weakens. If organic carbon burial declines, more of the oxygen generated by photosynthesis is consumed again. The system contains positive and negative feedbacks, each operating on different timescales. Phosphorus is especially important because cells cannot build genetic material, membranes, or energy transfer molecules without it. Reconstruction suggests its marine cycle was tightly coupled to oxygen during the Great Oxidation. Pulses of available phosphorus may have driven bursts of photosynthetic productivity and temporary oxygen increases. Later sequestration onto iron minerals may have helped suppress productivity. Scholars still debate the relative importance of these mechanisms, but nutrient supply clearly links the color of rocks to the activity of life. Phosphorus also reveals how alien these oceans remain. In some surface waters, phosphite may have represented a substantial portion of inorganic phosphorus. Modern oxygenated seas contain little of this reduced compound because it is readily oxidized. During the transition, however, microbial communities could exploit nutrient pathways that are now uncommon. Oxygen is spreading into a biosphere still structured by ancient chemistry, and for a long time both worlds coexist. Sulfur undergoes an equally profound reorganization. Greater oxidative weathering delivers more sulfate to the oceans. Microbes use sulfate in respiration, producing sulfide in anoxic waters and sediments. Where sulfide accumulates, it binds iron and creates euxinic conditions, waters that are both oxygen-free and rich in hydrogen sulfide. Such waters favor specialized anaerobes. Oxygenation can therefore increase the marine sulfate reservoir while indirectly helping sulfidic zones develop elsewhere. This creates a planet of sharp chemical contrasts. A shallow shelf may contain oxygen in its upper waters, iron-rich conditions below, and sulfidic mud at the bottom. The boundaries between them concentrate biological activity. Microbes oxidize methane where sulfate meets methane-rich water. Others cycle iron where reduced and oxidized minerals intersect. Cyanobacteria dominate the illuminated surface. Each layer exists because neighboring layers produce chemicals it can use. The continents also begin to influence climate differently. Oxidative weathering consumes oxygen, but can affect carbon dioxide through broader reactions involving silicate minerals and carbonate formation. Changes in continental area, rainfall, erosion, and tectonic uplift alter the rate at which these reactions proceed. Fresh rock exposed by glaciation or volcanic activity offers new surfaces for weathering. The planet's color, nutrient economy, atmospheric composition, and temperature become bound together in feedbacks with no single cause. Even the disappearance of dissolved iron from some surface waters changes the transparency and ecology of the ocean. Iron is both a reactive sink for oxygen and an essential nutrient. Removing it from solution may limit certain forms of photosynthesis in places where it becomes scarce. Elsewhere, upwelling or hydrothermal input restores the supply. Oxygen production can therefore destroy one of its own chemical supports by precipitating iron. Another example of life reshaping the conditions of its success. No forest marks this planetary transition. No animal migration records it. The visible monuments are mineral. Vast iron formations accumulate beneath ancient seas. Red sediments appear on land. Sulfate minerals become more common. Trace metals move through new pathways. Rocks that had once remained chemically reduced acquire oxidized coatings, while deeper environments preserve the older state. The world is rusting but not dying. Anaerobic ecosystems persist in sediments, subsurface rocks, and stagnant waters. Oxygenic life expands in illuminated zones. Between them lies a restless frontier where nutrients are released, trapped, recycled, and buried. The atmosphere's change is now written across continents and ocean basins, yet the pattern is uneven enough to resist a simple map. This is the visible face of the Great Oxidation, not a bright sky replacing darkness, but a planet divided by chemistry. Red land stands beside iron-rich seas. Oxygenated shallows overlook anoxic depths. Nutrients released by weathering fuel life, then vanish into minerals created by that same life. Earth's surface begins to resemble the oxidized world of the distant future, while most of its habitable volume still belongs to the ancient past. The new planet is emerging in color before it is emerging in air. The new planet is emerging in color before it is emerging in air, and its climate is beginning to feel the consequences. Methane has helped warm the young Earth beneath a sun that shines less brightly than the one known today. Much of that methane is biological, produced by methanogens in oxygen-free environments and released into the atmosphere where, under low oxygen conditions, it can survive long enough to become a powerful greenhouse gas. As oxygen spreads, that protection weakens. Oxygen does not simply replace methane molecule for molecule. It changes the chemistry that determines how long methane remains in the air. Methane reacts through pathways involving oxygen-derived radicals, and the growth of ozone alters how ultraviolet radiation penetrates the atmosphere. At the same time, oxygenated environments restrict the habitats where methanogens thrive and favour microbes that consume methane before it escapes from water or sediment. A greenhouse gas produced by the old anaerobic biosphere begins to lose both sources and atmospheric lifetime. The result may be severe cooling. Methane is far more effective than carbon dioxide at trapping heat over comparable concentrations, and on the early Earth it may have been central to maintaining liquid water under the faint young sun. If oxygen sharply reduces atmospheric methane while carbon dioxide is also drawn down by weathering and burial, the planet can cross a climatic threshold. Ice expands, brighter ice reflects more sunlight back to space. Cooling then strengthens itself through the ice albedo feedback. The Paleoproterozoic rock record preserves evidence for several major glaciations, often grouped under the name Huronian glaciations, beginning during the Great Oxidation. Glacial deposits appear across more than one continent. Drop stones, striated surfaces, diamictites, and other features indicate that ice reached places far from the poles. Some episodes may have approached a near-global frozen state, though scholars still debate how completely the oceans were sealed and whether the term snowball Earth should apply equally to every phase. The timing matters because oxygenation and glaciation are not perfectly simultaneous. Evidence suggests that an important rise in oxygen preceded at least one extreme Paleoproterozoic glaciation. This supports the idea that methane loss contributed to cooling, but it does not prove a single causal chain. Climate emerges from interacting systems, greenhouse gases, solar luminosity, continental position, weathering, volcanic carbon dioxide, ocean circulation, biological productivity, and the reflectivity of ice. Oxygen may pull one of the strongest levers without acting alone. Continents are arranged differently from today. Their exact positions remain uncertain because plate reconstructions become less reliable with great age. Yet their distribution affects climate profoundly. Land in tropical or subtropical regions can undergo intense chemical weathering, removing carbon dioxide from the atmosphere. Fresh volcanic provinces and tectonic uplift expose reactive rock. Rain carries weathering products to the ocean, influencing nutrient supply and carbon burial. If large areas of continental crust sit where warm, wet conditions accelerate weathering. The greenhouse can weaken before ice begins to spread. Once glaciation starts, the feedbacks become unstable. Ice covers land and shallow seas, reducing photosynthetic habitats and changing nutrient delivery to the ocean. Weathering slows beneath ice, which may eventually allow volcanic carbon dioxide to accumulate. Ocean circulation is reorganized. Biological productivity may decline in some regions while continuing in open water, beneath thin ice, or near hydrothermal systems. The biosphere contracts into refuges but does not disappear. For anaerobic life, the cold introduces a second pressure layered over oxygenation. Methanogens already losing territory near oxidized surfaces may persist in sediments, subglacial environments, and deep waters, Cyanobacteria face reduced light where ice and snow block the sun. Oxygen production may fall, yet oxygen consumption can also fall if productivity and respiration slow. The direction of atmospheric change depends on which processes weaken most. This is one reason the oxygen record appears to pulse rather than climb smoothly. A frozen planet is not chemically still. Beneath glaciers, rock is ground into fine sediment. Meltwater during retreats carries freshly crushed minerals toward the sea. When ice recedes more substantially, these reactive surfaces weather rapidly, releasing phosphorus and other nutrients. A post-glacial pulse of nutrient delivery can stimulate photosynthesis, increase organic carbon burial, and produce more oxygen. Cooling can therefore suppress the biosphere, prepare new mineral surfaces, and help trigger renewed oxygenation during recovery. Volcanic carbon dioxide offers another escape from deep freeze. Even if weathering nearly stops under extensive ice, volcanoes continue releasing carbon from Earth's interior. Over millions of years, carbon dioxide accumulates until greenhouse warming overcomes the reflectivity of the frozen surface. Deglaciation may then be abrupt on geological timescales. Melting ice lowers planetary albedo, warming accelerates, and oceans receive torrents of weathering products from newly exposed land. These transitions may help explain why oxygen rises and falls across the Great Oxidation Interval. Climate variability changes weathering rates, nutrient availability, productivity, methane cycling, and the balance between oxygen sources and sinks. Modeling published in 2024 suggests that such variability can generate multiple oxygenation episodes rather than one irreversible rise. A glaciation may reorganize the biosphere. Recovery may deliver phosphorus and stimulate oxygen production. Warmer conditions may then increase respiration and oxygen demand. The atmosphere oscillates as the planet repeatedly overshoots and corrects. The methane story also contains an important uncertainty. Ancient methane concentrations cannot be measured directly, and models depend on assumptions about biological production, hydrogen escape, haze formation, atmospheric chemistry, and volcanic gas fluxes. At very high methane relative to carbon dioxide, an organic haze might form, cooling the planet by reflecting sunlight even while methane acts as a greenhouse gas. Oxygen's influence on methane therefore occurs within a complex photochemical system, not a simple thermostat. No single explanation fully accounts for the Huronian glaciations. A decrease in methane is plausible and likely important, but continental weathering, reduced carbon dioxide, tectonic changes, and solar forcing all contribute. Even the number and duration of separate glacial episodes remain subjects of refinement as ancient rock sequences are redated and correlated. The surviving record is fragmentary because later tectonics, erosion, and metamorphism have destroyed much of the evidence. Still, the broad connection is difficult to ignore. Life changes atmospheric chemistry. Atmospheric chemistry changes climate. Climate then reshapes life and the processes producing oxygen. A waste gas released by cyanobacteria weakens the greenhouse maintained by anaerobic microbes, helping push Earth toward ice. The planet's first major oxygenation is therefore not only a chemical revolution, it may also be one of its earliest biologically amplified climate crises. Across frozen coastlines and darkened seas, the Old World endures in pockets, beneath ice, in sediments, and around hydrothermal vents. Microbial metabolisms continue. Above them, the atmosphere is no longer stable enough to preserve the ancient balance. Oxygen rises, methane fades, glaciers advance, and every change feeds back into the next. The sky has begun to turn against anaerobic life, and now the climate is following. The climate follows the atmosphere into instability, but the ancient biosphere does not vanish beneath ice or oxygen. It withdraws. The safest places are no longer the open surface waters where anaerobic metabolisms once spread widely. They are the places oxygen reaches slowly, weakly, or not at all mud beneath the seafloor, stagnant basins, pores inside rock, hydrothermal systems, and the interiors of layered microbial communities. The old world survives by moving downward. In sediments, oxygen is consumed rapidly, organic matter settles from the water above, and microbes begin breaking it down. Where oxygen is present, aerobic respiration uses it first because it yields abundant energy. Once the oxygen disappears, other metabolisms take over in a sequence shaped by chemical opportunity. Nitrate may be used, then manganese and iron compounds, then sulfate. Deeper still. Methanogens produce methane from carbon dioxide, hydrogen, acetate, and other substrates. A few centimeters of mud can contain a vertical history of Earth's atmosphere, from an oxygenated surface to conditions resembling the distant Archean. This layered organization is not merely a remnant of the past, it is a survival mechanism. Microbes near the sediment surface consume incoming oxygen and shield organisms below. Sulfide produced at depth meets oxygen or oxidized metals near the boundary and is transformed before spreading upward. Methane rising from deeper layers is consumed by microbial partnerships where methane and sulfate overlap, reducing how much reaches the ocean and air. The habitat is divided into narrow chemical territories, each maintained by the metabolism of its neighbors. As surface oceans become more oxygenated, these boundaries descend. Anaerobic organisms follow them into deeper sediments or waters isolated from circulation. Restricted basins become refuges where oxygen delivered from above is exhausted before reaching the bottom. Deep ocean regions remain anoxic for long intervals because atmospheric oxygen is still low and ventilation is weak. In some places, dissolved iron dominates. In others, sulfide accumulates. The Great Oxidation changes the upper world first, leaving much of the ocean interior chemically ancient. Hydrothermal systems provide another sanctuary. Fluids emerge carrying hydrogen, sulfide, methane, ferrous iron, and other reduced compounds. These chemicals support ecosystems that do not depend directly on sunlight. Oxygen may penetrate the outer edges of such systems and create new metabolic interfaces, but deeper fluids and subsurface fractures remain reducing. The same geology that once supplied oxygen sinks, now protects organisms harmed by oxygen. The continental subsurface offers even greater isolation. Water moves through cracks and pores, reacting with minerals and carrying dissolved gases. Microbes survive at depths where sunlight has never reached, and atmospheric change arrives only through slow geochemical pathways. Some draw energy from hydrogen produced when water reacts with iron-bearing rock. Others use sulfur, carbon compounds, or radiolytically generated chemicals. Their growth may be extremely slow, yet persistence matters more than speed. Modern drilling and mine studies reveal that such deep microbial worlds still exist. Organisms inhabit oceanic crust, buried sediments, aquifers, and fractures kilometers below the surface. They are not unchanged Archean fossils, and their lineages have evolved for billions of years. But many rely on metabolisms rooted in the oxygen-poor past. Their presence shows that atmospheric oxygenation never eliminated anaerobic life. it redistributed it into a vast hidden biosphere. Even at the modern surface, anaerobic refugees remain common. Wetland mud, lake sediments, waterlogged soils, sewage, and oxygen-starved marine zones support communities that cannot tolerate prolonged oxygen exposure. Methanogens still produce methane. Sulfate reducers still generate sulfide. Fermenters still break organic molecules into compounds used by other microbes. The ancient metabolic web survives inside the oxygenated world because local consumption can remove oxygen faster than it arrives. This persistence changes how the Great Oxidation should be understood. A planetary atmosphere can become hostile without making every habitat hostile. Oxygen reaches environments unevenly. Its penetration depends on diffusion, mixing, temperature, organic matter, mineral reactivity, and biological consumption. A millimeter of microbial activity can separate an oxygenated zone from an anoxic one. At planetary scale, entire ocean basins can remain reducing beneath an oxidized sky. Refuge, however, comes with costs. Habitats may become smaller, more isolated, and less productive. Nutrient flow changes when oxygen alters iron and phosphorus cycling. Organisms confined to sediments depend on organic material arriving from above or on slow geological energy sources. Populations separated by oxygenated barriers may diverge or disappear locally. The fossil record cannot count these losses, but ecological contraction almost certainly accompanies survival. Some lineages respond by becoming facultative, able to use oxygen when it's present and switch to other pathways when it's absent. Others retain strict anaerobic metabolisms, but develop enzymes that tolerate brief encounters with reactive oxygen. Communities cooperate across redox boundaries, with one organism consuming what another produces. Horizontal gene transfer can spread protective or metabolic tools. Survival is not passive retreat alone, it is biochemical negotiation with a changing planet. The deep ocean remains especially important because oxygenation there lags far behind the atmosphere. Persistent oxygen in surface waters does not guarantee ventilation at depth. Ocean circulation depends on temperature gradients, salinity, basin shape, and climate. During parts of the Paleoproterozoic, deep waters likely remain ferruginous, rich in dissolved iron, while some regions become euxinic and sulfide-rich. These environments support anaerobic ecosystems long after the Great Oxidation has begun. The boundary between oxygenated and anoxic water can also become a zone of intense productivity. Microbes gather where opposing chemicals meet. Iron oxidizers exploit ferrous iron rising toward oxygen. Sulfur oxidizers use sulfide moving upward from anoxic depths. Methane oxidizers intercept methane before it escapes. Each group depends on a gradient created by planetary disequilibrium. Oxygen does not simply replace old metabolisms, it creates interfaces where old and new metabolisms become interdependent. Scholars still debate how extensive these refugees were and how the distribution of ocean chemistry changed through each oxygenation pulse. Ancient sediments sample only particular basins, and later, alteration can blur the original signal. Yet no plausible reconstruction produces a uniformly oxygenated planet. The evidence instead points to long coexistence oxidized continents and anoxic depths, oxygenated shallows and reducing mud, ozone above and methanogenesis below. That coexistence becomes one of Earth's enduring patterns. Oxygen rules the open atmosphere, but anaerobic life persists wherever chemistry permits it. The old biosphere is compressed rather than erased, hidden beneath surfaces increasingly shaped by its own descendants and competitors. In every dark sediment where oxygen disappears, the planet briefly returns to an earlier state. The retreat therefore carries a quiet legacy. Life does not survive because Earth remains unchanged. It survives because the planet is heterogeneous, filled with boundaries, shelters, and slow pathways. Beneath the altered sky, ancient metabolisms continue in darkness, waiting at the edges of oxygen's reach. The world above is becoming unfamiliar, but below it, the chemistry of the first living Earth is never entirely lost. The refuges preserve the ancient metabolisms, but elsewhere life begins to do something more radical than hide from oxygen. It learns to control it. What entered the biosphere as reactive waste becomes a source of chemical power, provided cells can keep the reaction from destroying them. This change is not a single invention. It is a long accumulation of defenses, enzymes, membranes, and partnerships that allow organisms to survive near oxygen, then tolerate it, and finally depend upon it. The challenge is protection. Oxygen itself is reactive, but some of its most dangerous effects come from unstable derivatives such as superoxide, hydrogen peroxide, and hydroxyl radicals. These can damage proteins, alter membranes, and injure genetic material. Cells exposed to even small amounts of oxygen need ways to neutralize them. Enzymes such as superoxide dismutase, catalase, and peroxidases perform this work in modern organisms, converting dangerous molecules into less harmful products. Their evolutionary histories are complex, and scholars still debate when particular forms first appeared, but some protective systems almost certainly arose before atmospheric oxygen became abundant. Protection alone does not make oxygen useful. To gain energy from it, cells must connect oxygen reduction to an electron transport chain. Electrons removed from food or other chemical donors pass through membrane-bound proteins. Their movement pumps protons across a membrane, storing energy as an electrochemical gradient. The return flow powers ATP synthase, a molecular rotary machine that produces ATP, the cell's immediate energy currency. Oxygen receives the electrons at the end of the chain and combines with protons to form water. This arrangement is remarkably efficient. Fermentation and many anaerobic pathways recover only a limited portion of the energy stored in organic molecules. Aerobic respiration can extract far more. The difference is large enough to change what biological systems can support. More available energy can fund active transport, molecular repair, larger genomes, complex regulation, and faster growth when nutrients permit. Oxygen does not automatically create complexity, yet it expands the energetic possibilities from which complexity can evolve. Evidence from molecular clocks suggests that some hemicopper oxygen reductases, enzymes used in oxygen respiration, may have diversified between roughly 3.2 and 2.8 billion years ago. If these estimates are broadly correct, oxygen use began in localized Archean environments long before the Great Oxidation Event. Microbes living around oxygen oases did not wait for the atmosphere to change. They exploited tiny, fluctuating supplies produced by nearby photosynthesizers. That early adaptation adds another twist to oxygen's delayed rise. Oxygen-consuming organisms become a biological sink. Cyanobacteria release oxygen, and neighboring respirers immediately use it to oxidize organic matter or reduced chemicals. The more successful oxygen production becomes locally, the more opportunity it creates for oxygen users to expand. Life therefore develops a feedback that consumes the gas later associated with planetary transformation. The road to an oxygenated atmosphere passes through ecosystems increasingly effective at preventing oxygen from escaping. Competition intensifies along the redox frontier. Organisms able to tolerate oxygen can approach the sunlit surface, where photosynthetic production is high. Strict anaerobes remain below. Aerobic heterotrophs consume organic matter where oxygen is available, while fermenters and methanogens process what reaches deeper layers. Other microbes use nitrate, manganese, iron, or sulfate. The community becomes a ladder of electron acceptors arranged by energetic reward and chemical availability. Oxygen usually sits near the top of that ladder. Its strong tendency to accept electrons allows organisms to release large amounts of energy from reduced compounds. Some microbes oxidize hydrogen with oxygen. Others use iron, sulfur, methane, or carbon monoxide. Oxygen respiration is not limited to consuming organic food. It becomes attached to many geochemical metabolisms. As the gas spreads, it links atmospheric change to the cycling of elements throughout the ocean and crust. Yet oxygen use remains dangerous even for organisms that depend upon it. Aerobic life constantly repairs oxidative damage. Cells regulate oxygen delivery, sequester reactive metals, and maintain antioxidant systems. The harmony between life and oxygen is a controlled tension, not complete safety. Every breath in a later animal will depend on molecules whose reactivity must be carefully contained. The expansion of oxygen respiration may also transform ecological efficiency. Organic matter that once passed through long anaerobic chains can be oxidized more completely near oxygenated surfaces. This reduces the amount reaching methanogens and can further suppress methane emissions. It also recycles nutrients rapidly, potentially supporting greater productivity while increasing oxygen demand. Whether oxygen rises depends not simply on how much photosynthesis occurs, but on how much organic carbon escapes this efficient respiratory recycling and becomes buried. Some steps may occur through cooperation rather than competition. Genes for oxygen defenses and respiration can move between microbial lineages through horizontal transfer. A useful enzyme does not always remain confined to the descendants of the cell in which it first evolved. Microbes exchange genetic material, allowing innovations to spread across branches of life. The transition may therefore produce a web of adaptation rather than a single lineage marching toward the future. The distinction between the major domains of life also matters. Bacteria and archaea contain enormous metabolic diversity, including oxygenic photosynthesis, aerobic respiration, methanogenesis, sulfur cycling, and many pathways still unfamiliar outside microbiology. The later emergence of eukaryotic cells will involve another transformation in energy management, but the foundations are microbial. Long before plants or animals, prokaryotic communities invent the chemical systems that make an oxygenated biosphere possible. No fossil records the first cell that breathed oxygen. Enzymes leave no skeletons, and molecular clocks carry uncertainties in evolutionary rate and gene transfer. Geological evidence can reveal when oxygen affected environments, but not necessarily which organisms used it there. What survives in the record suggests a prolonged period of experimentation with oxygen production, detoxification, and respiration evolving in overlapping stages. The contrast between oxygen as poison and life-giving gas is too simple. It is both from the beginning. Its danger creates selection for protection. Protection permits exposure. Exposure creates opportunity for respiration. Respiration yields more energy but also consumes oxygen and produces new ecological pressures. Each adaptation changes the environment in which the next one evolves. By the later phases of the Great Oxidation, organisms capable of using oxygen are no longer confined to brief daytime oases. Persistently oxygenated surface waters provide expanding territory. Aerobic metabolisms become integrated into marine nutrient cycles and food webs. Anaerobes remain dominant in deeper waters and sediments, but the surface biosphere increasingly runs on a gas once treated only as contamination. Life has not defeated oxygen. It has domesticated a portion of its reactivity. The achievement is incomplete, costly, and never entirely safe, but it changes the future of the planet. A substance that narrowed ancient habitats now opens an energetic landscape unavailable to the old biosphere. The atmosphere turned against many forms of anaerobic life, yet evolution answered by making the atmosphere part of metabolism itself. If oxygen has become a durable feature of the surface world and life has learned to use it, there is an obvious temptation to imagine the rest of the story unfolding quickly. The atmosphere rises, the oceans clear, and the stage is set for complex life. But the geological record denies that clean progression. After the Great Oxidation, oxygen does not continue climbing steadily toward modern abundance. Instead, Earth seems to settle into a long and unstable middle state, one in which the surface is more oxidized than before, but much of the ocean and crust remain profoundly unlike the world to come. This prolonged interval is sometimes described as a period of low but persistent oxygen, a kind of planetary pause. The atmosphere contains enough oxygen to sustain oxidative weathering, suppress the old sulfur isotope signature, and maintain at least some ozone, yet likely far less than 1% of the present atmospheric level for long spans. Estimates remain uncertain and depend on proxy interpretation. But the broad conclusion is robust. The Great Oxidation marks the beginning of an oxygenated surface Earth, not the arrival of a modern one. The oceans reveal this incompleteness especially clearly. Surface waters in some regions retain oxygen, but deep waters remain ferruginous, rich in dissolved ferrous iron, or become euxinic, rich in hydrogen sulfide. These conditions constrain where many organisms can live and how nutrients cycle. Iron and sulfide influence trace metals such as molybdenum, copper, and zinc, which are needed by various enzymes. A biosphere living under low oxygen may therefore be limited not simply by the gas itself, but by the availability of catalytic metals and the chemistry that delivers them. This helps explain why oxygenation can stall. As the surface environment becomes more oxidized, phosphorus may be trapped more efficiently by iron minerals, reducing nutrient availability for photosynthetic organisms. If primary productivity weakens, oxygen production slows. At the same time, organic matter reaching anoxic deep waters can be decomposed through sulfate reduction and other pathways that do not require free oxygen, returning carbon to the system without contributing to an oxygen rise. The balance between source and sink remains delicate. Volcanic and metamorphic gases continue to matter as well. Even if the mantle slowly becomes less reducing through geological time, Earth's interior remains a source of reduced compounds capable of consuming oxygen. Tectonic processes alter the ratio of volcanic carbon dioxide to more reducing species, while continental growth changes the area available for weathering and nutrient delivery. The Great Oxidation does not end geochemical resistance. It merely weakens the oldest equilibrium enough for a new one to emerge. The biosphere itself can also maintain the low oxygen state. Aerobic respiration in surface environments becomes efficient at consuming organic matter before it is buried. If recycling strengthens faster than carbon burial, atmospheric oxygen may plateau. Productive shallow seas can produce oxygen during growth and consume much of it again during decomposition. A world with active photosynthesis and widespread oxygen use does not necessarily become highly oxygenated. It can stabilize in a tight cycle where oxygen is continually produced and continually spent. Recent work placing the Great Oxidation within a much longer 2 billion year surface oxygenation transition reinforces this view. Earth does not move from an anoxic planet to a modern oxygen world in one leap. It enters a drawn-out middle chapter. The atmosphere has crossed a threshold, but the deep ocean, nutrient cycles, and geological sinks keep pulling against further change. In this sense, the Great Oxidation solves one planetary problem while creating another. How can oxygen continue rising once it has already altered the systems that first allowed it to appear? There is also a biological paradox. The new energetic advantages of oxygen may encourage more productive ecosystems, but productive ecosystems also generate more organic matter to be decomposed, potentially increasing oxygen demand. Meanwhile, anaerobic processes remain strong in deeper waters and sediments, producing methane and sulfide where conditions allow. Old and new metabolisms coexist in tension. The planet is neither anaerobic in the Archean sense nor oxygen-rich in the later Phanerozoic sense. It is intermediate, chemically stratified, and stubbornly unresolved. Scholars sometimes refer to later intervals, especially the mid-Proterozoic, as a boring billion, though the phrase can mislead. The chemistry is not boring. It is subtle, slow, and resistant to dramatic simplification. Low oxygen conditions may have constrained the timing of eukaryotic diversification and limited ecological complexity, but they also sustained specialized microbial ecosystems and intricate redox boundaries across vast marine regions. A world can be stable for hundreds of millions of years and still remain dynamically balanced on geological feedbacks. The stall also forces humility in interpreting the Great Oxidation itself. If oxygen rose once and then remained limited for so long, then whatever drove the initial transition was not sufficient to complete the transformation. The causes must be multiple and the thresholds different. A change that allows oxygen to persist in the atmosphere is not the same as a change that oxygenates the deep sea, and neither guarantees the nutrient conditions needed for later biological expansion. Each step requires its own explanation. Proxy records from this long aftermath are difficult to read. Later burial, metamorphism, and erosion erase much of the evidence. Marine chemistry varies from basin to basin, making local records hard to scale globally. Different proxies record different thresholds. Some indicate oxic surface conditions, others suggest persistent deep anoxia. The uncertainty is frustrating, but it is also revealing. Earth's surface is not moving along a simple one-dimensional path. It is occupying different oxidation states in different compartments at the same time. For anaerobic life, this prolonged middle state is a reprieve as much as a defeat. Surface conditions have narrowed its range, yet vast marine and subsurface habitats remain available. For oxygen-using organisms, the world is promising but constrained. More energy is available than before, but oxygen remains too limited and unevenly distributed to remake the entire biosphere at once. The future is possible without yet being inevitable. The red beds, sulfur signals, and oxygenated shallows therefore do not mark the end of Earth's atmospheric crisis. They mark the beginning of a long negotiation. The atmosphere has turned against the old surface world, but it has not committed fully to the new one. Oxygen hangs at low levels, sufficient to transform weathering and ecology, insufficient to modernize the entire ocean. The planet lingers in this suspended condition, because every gain in oxygen activates sinks, nutrient traps, and recycling pathways that keep it from rising freely. This long stagnation matters because it preserves the central truth of the story. Life does not simply invent oxygen and move on. It struggles with its consequences for an extraordinary stretch of time. The Great Oxidation is not a triumphant opening act followed by smooth progress. It is the beginning of a slow planetary argument between production and consumption, surface and depth, innovation and resistance. That argument will eventually shape the conditions for complex cells and larger organisms, but not before the Earth spends eons learning how to live with the gas it created. The planet's long argument with oxygen continues, but somewhere within that drawn-out middle state, a new kind of cell begins to change the possibilities of life. Eukaryotes are organisms whose cells contain a nucleus and complex internal structures. Every animal, plant, fungus, and many single-celled organisms belong to this domain. Their emergence is one of the great transitions in biological history, yet the relationship between oxygen and their origin remains more complicated than any simple statement that oxygen caused complex life. The earliest evidence for eukaryotes is fragmentary and debated. Molecular clocks often place their origins deep in the Proterozoic, while fossils and chemical biomarkers provide a less complete and sometimes contested record. Some structures once interpreted as early eukaryotic fossils have later been reclassified, and some biomarkers can be affected by contamination or produced through unexpected pathways. What survives suggests that eukaryotic lineages were present long before animals, but their exact timing, diversity, and ecological importance remain uncertain. A eukaryotic cell differs from a bacterium or archaeon not only in size, it contains compartments that separate chemical processes, a dynamic internal skeleton, complex systems for moving material, and often a much larger genome. These features require energy, coordination, and regulation. Oxygen may help support such complexity because aerobic respiration yields large amounts of ATP, yet oxygen alone cannot explain the origin of the eukaryotic cell. Many bacteria use oxygen efficiently without becoming eukaryotes, while some modern eukaryotes survive in low oxygen or anoxic environments. The central event is endosymbiosis. At some point, an archaeal host forms a lasting partnership with a bacterium capable of oxygen-based respiration. Instead of being digested or expelled, the bacterium remains inside the host and eventually becomes the mitochondrion. Modern mitochondria still carry traces of that bacterial ancestry in their membranes, genetic material, and molecular machinery. This merger joins two evolutionary histories and creates a cell able to distribute respiratory power through internal compartments. How that partnership began remains disputed. It may have started as predation, metabolic exchange, syntrophy, or another form of close association. Scholars still debate whether the host already possessed complex internal structures before acquiring the mitochondrial ancestor, or whether the energy and genes supplied by the symbiont enabled much of that complexity afterward. The fossil record does not preserve the moment, and modern cells are the descendants of countless later changes. Oxygen gives the partnership an obvious advantage in environments where it is available. A cell containing many mitochondria can place respiratory membranes throughout its interior, instead of relying only on the outer cell membrane. This arrangement may allow greater energy production as the cell grows larger. More energy can support protein synthesis, genome maintenance, active transport, internal trafficking, and the regulation of thousands of genes. But this energetic explanation remains a hypothesis about evolutionary opportunity. not proof that low oxygen alone prevented complex cells from appearing earlier. The timing also resists a neat connection. The Great Oxidation begins more than 2 billion years ago, while clear evidence for abundant and ecologically important eukaryotes becomes much stronger much later. If oxygen were the only missing ingredient, the delay would be difficult to explain. Low and unstable oxygen may have imposed constraints, but nutrient availability, trace metals, ecological competition, genome evolution, predation, and the rarity of successful endosymbiosis may all have mattered. The oceans inhabited by early eukaryotes are chemically divided. Oxygenated surface waters occupy some regions, while deeper layers remain ferruginous or sulfidic. A cell capable of aerobic respiration may flourish near the surface, but encounter severe limits below. Sulfide can interfere with respiratory enzymes and alter the availability of metals needed for metabolism. Iron-rich waters create different opportunities and hazards. Eukaryotes must navigate not one global oxygen level but shifting mosaics of light, nutrients, oxygen, iron, and sulfur. Some early forms may remain small and single-celled, grazing on bacteria or absorbing dissolved nutrients. Others develop flexible membranes that allow them to engulf particles and prey. Phagocytosis, the ability to surround and internalize another cell, may transform ecology by introducing active microbial predation on a larger scale. Once one cell can consume another, defensive structures, larger body size, motility, and new food web relationships can become advantageous. Whether phagocytosis preceded mitochondrial acquisition or followed it remains part of the debate over eukaryotic origins. Oxygen may influence this ecological expansion indirectly. Aerobic respiration can support active movement and complex feeding, but oxygenation also changes the abundance and distribution of bacterial prey. It alters nutrient cycles, trace metal chemistry, and the preservation of organic matter. A modest rise in oxygen can reorganize entire food webs without producing anything close to a modern atmosphere. Complex cells evolve inside their changing network, not outside it. There is a further irony. Eukaryotes inherit both the benefits and hazards of oxygen. Mitochondria generate ATP by transferring electrons toward oxygen, but that process can also produce reactive oxygen species. Eukaryotic cells must maintain antioxidant defenses, repair damaged molecules, and regulate mitochondrial activity. Programmed cell death may even use mitochondrial signals, turning the organelle that powers the cell into part of its self-destruct machinery. The old poison is not eliminated, it is enclosed, managed, and made useful. Some eukaryotic lineages later adapt to environments with little or no oxygen. Their mitochondria become modified into hydrogenosomes or mitosomes, organelles that no longer perform conventional aerobic respiration. These organisms show that eukaryotic complexity does not require constant exposure to oxygen once the cellular architecture exists. Their ancestors, however, almost certainly possessed mitochondria, suggesting that the foundational symbiosis occurred in a world where oxygen mattered, even if it was unevenly distributed. The appearance of eukaryotes, therefore, does not mark the immediate triumph of complex life. For long periods, they may remain minor participants in microbial ecosystems dominated by bacteria and archaea. Their fossils are rare, their diversity difficult to measure, and their environmental range limited by ocean chemistry. The evidence remains incomplete, and no single oxygen threshold can be identified as the moment complexity becomes possible. What oxygen provides is not destiny but room. It enlarges the energetic landscape, creates new ecological interfaces, and favors cells capable of controlling reactive chemistry. Endosymbiosis then converts that opportunity into a new cellular design. The result is a lineage with extraordinary future potential, but the future remains distant. Animals do not appear, plants do not cover land, and the oceans are still largely anoxic at depth. The long oxygen stagnation is therefore not empty time. Within it, life experiments with scale, partnership, predation, compartmentalization, and energy. The atmosphere has already ended the unquestioned dominance of the old anaerobic surface world. Now, inside the patchwork conditions left behind, cells begin assembling the architecture from which later complexity will grow. Oxygen has not created the eukaryotic cell by itself, but without the new redox world, that cell's possibilities may have been far narrower. The first rise of oxygen changes the direction of Earth's history, but it does not finish the transformation. For hundreds of millions of years after the Great Oxidation, the atmosphere remains thin in oxygen, the deep ocean stays largely anoxic, and complex cells occupy only part of the microbial world. The planet has crossed one threshold and then stalled before another. A second atmospheric awakening will require a different combination of biology, geology, and time. This later transition does not belong to a single date. It unfolds through the Neo-Proterozoic, beginning more than 1 billion years after the Great Oxidation, and intensifying across several pulses between roughly 800 and 540 million years ago. Some researchers describe this as the Neo-Proterozoic Oxygenation Event, though the name can imply more unity than the evidence supports. Oxygen appears to rise in stages, with reversals, while ocean chemistry responds unevenly. Continental growth and tectonic reorganization alter the setting. Supercontinents assemble and break apart. Mountain building exposes fresh rock. Rivers carry larger loads of weathering products toward the sea. New coastlines and broad continental shelves create shallow marine habitats where sunlight, nutrients, and circulation can support high productivity. The exact importance of each tectonic episode remains debated, but the geography of the planet is becoming more favorable to sustained exchange between land, ocean, atmosphere, and life. Weathering releases phosphorus and other nutrients, stimulating photosynthesis in some regions. More organic matter is produced, and where burial outpaces decomposition, oxygen can accumulate, yet the same process can increase respiration and oxygen demand. The system still contains breaks. Iron minerals capture phosphorus. Sulfidic waters remove trace metals. Reduced volcanic gases and exposed crust consume oxygen. A lasting rise requires several sinks to weaken or become overwhelmed. The biological community is also different now. Eukaryotic algae diversify and spread through the oceans, adding larger cells and new ecological strategies to photosynthetic ecosystems. Some algae sink more efficiently than cyanobacteria, carrying organic carbon into deeper water and sediment before it can be fully recycled near the surface. This may increase the effectiveness of the biological carbon pump, though its timing and magnitude remain uncertain. A change in who performs photosynthesis can alter how much oxygen escapes immediate consumption. Predation may contribute as well. Larger eukaryotic cells consume smaller organisms, package organic matter into particles, and change the structure of marine food webs. Waste pellets, aggregates, and dead cells sink, transporting carbon away from oxygenated surface waters. The deeper this material travels before decomposition, the more likely some fraction is to be buried, ecology becomes part of atmospheric regulation. Oxygen levels depend not only on how much life grows, but on what eats it, how particles form, and where decay occurs. Meanwhile, the oceans remain chemically divided. Oxygenated surface waters expand in some regions, but deep basins can still be ferruginous or euxinic. Trace metals needed for enzymes become available in one place and scarce in another. Molybdenum, copper, zinc, and iron circulate through redox-sensitive pathways. These elements matter because complex cells require sophisticated enzymes for respiration, nitrogen cycling, and antioxidant defense. Rising oxygen can improve access to some metals while removing others from solution, creating a shifting chemical landscape rather than universal enrichment. The Neoproterozoic is also marked by glaciations, including the Sturtian and Marinoan episodes, when ice may have reached tropical latitudes. These later snowball Earth events are separated from the Huronian glaciations by more than 1 billion years, and their causes are not identical. Continental position, carbon dioxide drawdown, weathering, and ice albedo feedbacks all contribute. Their relationship to oxygenation is complex. Glaciation can suppress productivity, alter ocean circulation, and isolate habitats, but deglaciation can deliver enormous pulses of nutrients from freshly weathered rock. When the ice retreats, rivers carry phosphorus, iron, and other materials into the ocean. Greenhouse conditions following deglaciation may accelerate weathering. Photosynthetic productivity can rise, organic carbon burial may increase, and oxygen production may strengthen. Some models connect these post-glacial nutrient pulses to later oxygenation and biological expansion. The evidence remains incomplete, and no single explanation fully accounts for the sequence. But climate extremes again appear entangled with atmospheric change. By the late Neoproterozoic, oxygenated zones seem to spread through shallow seas. Deep waters remain inconsistent, yet the habitable volume for oxygen-dependent organisms increases. Larger bodies become easier to sustain where oxygen can diffuse through tissues. Active movement, predation, and complex development become more energetically feasible. Oxygen does not dictate anatomy, but it places limits on how large and active an organism can become without specialized systems for transport. This changing world is inhabited by the Ediacaran biota, a diverse collection of large, soft-bodied organisms appearing before the Cambrian explosion. Some resemble fronds, quilted discs, or branching forms unlike most later animals. Others may represent early branches of animal life, while some have no clear modern relatives. Their biological identities remain debated, but their presence shows that macroscopic multicellular organisms were establishing themselves in marine ecosystems before abundant skeletons appear. The connection between oxygen and the Ediacaran biota is not simple. Some organisms may tolerate low oxygen, and local oxygenated settings can exist even when global levels remain modest. Nutrient availability, ecology, developmental genetics, and environmental stability all matter. It would be misleading to imagine that oxygen rises and animals immediately appear. Instead, expanding oxygenated habitats remove one constraint, while other innovations determine how life responds. A further rise occurs near the Ediacaran-Cambrian transition. More persistent oxygenation, ecological escalation, and the spread of burrowing organisms begin to transform the seafloor. Animals disturb sediment, mix oxidants downward, and alter nutrient recycling. This bioturbation changes the chemistry of marine mud and may further influence oxygen demand. Once again, Life does not merely respond to the environment, it modifies the system that sustains it. The second awakening is therefore a feedback between planet and biosphere. Tectonics creates new continents and shelves. Weathering supplies nutrients. Algae and food webs alter carbon export. Organic burial leaves oxygen behind. Oxygen expands habitable space for active multicellular organisms. Those organisms then rework sediments and reshape chemical cycles. Each step opens possibilities while creating new sinks and constraints. By the dawn of the Cambrian, Earth still does not possess uniformly modern oceans. Anoxic waters persist, and oxygen levels remain uncertain. Yet the surface world has changed enough to support larger, mobile, increasingly complex organisms across expanding marine environments. The long stagnation has begun to break. The first oxidation ended the permanence of the anaerobic surface world. The second enlarges the realm in which oxygen can power complexity. Between them lies more than 1 billion years of hesitation, experimentation, and chemical resistance. Earth's atmosphere does not awaken once. It awakens in stages, each one reshaping the ocean, widening the energy available to life, and preparing the planet for forms that could never have survived beneath its earlier sky. The second atmospheric awakening shows what oxygen eventually made possible, but it can obscure the deeper mystery of the first crisis. What happened to life when the atmosphere began to turn against anaerobic ecosystems? The answer is more complex than a simple story of mass poisoning. There is no fossil ledger recording the number of microbial lineages lost. There are no bones marking a single horizon of death. Instead, scientists must infer biological disruption from changes in productivity, nutrient cycling, isotope ratios, mineral deposits, and the shrinking geography of low-oxygen habitats. One explanation emphasizes direct toxicity. Oxygen and its reactive derivatives damage cells that lack protective enzymes. As oxygenated waters spread across shallow seas, strict anaerobes would have been forced into deeper sediments, restricted basins, or subsurface refuges. Local populations likely vanished where exposure persisted, yet direct poisoning cannot explain every feature of the record. Oxygen levels remained extremely low by modern standards. Much of the ocean stayed anoxic, and anaerobic life had many places to survive. The evidence supports displacement and local extinction, not near erasure of the biosphere. A second explanation focuses on methane. Methanogens helped maintain a greenhouse atmosphere under the faint young sun. Rising oxygen reduced methane production in exposed environments, encouraged methane-consuming microbes, and altered reactions that controlled methane's lifetime. Cooling followed, perhaps contributing to the Huronian glaciations. In this model, oxygen harms the old biosphere indirectly by weakening the climate system it helped create. Ice reduces open water habitat, changes circulation, limits light, and reorganizes nutrient delivery. But methane loss is not the only plausible driver of glaciation. Carbon dioxide drawdown, continental weathering, tectonic geography, volcanic fluxes, and ice albedo feedbacks also matter. A third explanation centers on nutrients. Phosphorus can stimulate photosynthesis and oxygen production. Yet oxygenated iron minerals can also remove phosphorus from seawater. During and after the Great Oxidation, this tug-of-war may have produced bursts of productivity followed by scarcity. If phosphorus became less available, primary production could fall sharply, reducing the biological energy entering marine ecosystems. A study of the sulfate isotopes near the end of the Great Oxidation has been interpreted as evidence for a major decline in gross primary productivity. Popular accounts sometimes convert that decline into a claim that most life became extinct. The rocks do not justify that conversion. Productivity is not the same as species diversity, and an isotope-based estimate is not a direct census of organisms. The proposed productivity collapse may still represent a profound ecological event. Less photosynthesis means less organic matter for microbial food webs, less oxygen production, and fewer resources reaching deeper communities. If the decline followed enhanced nutrient delivery and biological expansion, the contrast could have been severe. A larger biosphere may have contracted when weathering slowed, phosphorus was trapped, or post-glacial conditions changed. Yet the timing, magnitude, and global extent remain debated. Different basins could respond differently, and the sedimentary record is too incomplete to trace every ecosystem. Another possibility is that the biosphere did not simply shrink. It may have grown in total productivity during parts of the transition while changing composition dramatically. Oxygenic photosynthesis opens access to water as an electron source, and aerobic respiration extracts more energy from organic matter. These innovations can support greater biological activity even while many anaerobic habitats contract. A 2026 reassessment argues that the Great Oxidation should not automatically be treated as the biological catastrophe implied by popular narratives. The atmosphere forced deep reorganization, but the total biosphere may have expanded during portions of the event. This apparent contradiction dissolves when scale is considered. A planet can become more productive overall while particular ecosystems collapse. Cyanobacteria may spread across sunlit waters as methanogenic communities lose territory. Oxygen-using organisms may flourish along new redox boundaries while strict anaerobes retreat. More carbon may be fixed globally, yet fewer nutrients may reach certain basins. The Great Oxidation can be both opportunity and crisis, depending on which organism, habitat, and interval is examined. Climate variability adds another layer. Models suggest that transitions between warmer and colder states could drive repeated oxygenation episodes. Glacial advance suppresses weathering and alters productivity. Deglaciation exposes finely ground rock, releasing nutrients and stimulating photosynthesis. Oxygen rises, methane falls, and cooling may return. Such feedbacks can produce oscillations rather than a one-way transition. They also mean biological stress may arrive in waves, separated by periods of recovery or expansion. No single catastrophe horizon is required. Geology itself can change the balance independently of biology. The oxidation state of volcanic gases, the growth of continents, the burial of organic carbon, and the supply of reduced iron all evolve through time. A shift in mantle or crustal processes might reduce the planet's oxygen demand, allowing atmospheric accumulation without a sudden biological innovation. Conversely, renewed volcanic emissions or exposure of reduced minerals could pull oxygen downward again. The Great Oxidation reflects competition between life and the solid Earth, not biology acting alone. Photochemistry may provide threshold behavior. Once enough oxygen exists to form ozone, ultraviolet radiation and sulfur chemistry change. Methane lifetime changes. Atmospheric escape processes shift. Small increases in oxygen may then produce disproportionately large effects, moving the atmosphere into a different stable state. Similar threshold models are now explored for exoplanets, where oxygen might accumulate through feedbacks rather than gradual change. Even so, a photochemical tipping point cannot explain nutrient records, glacial deposits, or biological adaptation by itself. Every explanation captures part of the event. Direct toxicity explains why anaerobic habitats shrink. Methane loss connects oxygen to cooling. Nutrient limitation helps account for productivity changes. Organic carbon burial explains how oxygen can remain behind. Volcanic evolution affects the strength of oxygen sinks. Climate oscillations explain repeated rises and falls. Aerobic metabolism explains why the new gas becomes an opportunity rather than only a threat. The difficulty is that these plausible mechanisms interact. The surviving record preserves outcomes more clearly than causes. Red beds show oxidation on land. Sulfur isotopes reveal atmospheric thresholds. Trace metals record changing marine redox conditions. Glacial sediments show climate extremes. Molecular clocks suggest oxygen use before global oxygenation. None of these archives captures the entire system, and correlations between them do not always prove causation. Scholars still debate the ordering and relative strength of the feedbacks. The safest conclusion is also the most compelling. Earth's first oxygen crisis was not a single extinction event, a single climate disaster, or a single biochemical breakthrough. It was a prolonged reorganization in which atmosphere, ocean, rock, and life repeatedly forced one another into new states. The anaerobic world lost its control of the surface, but it did not disappear. Oxygen-using life gained power, but it did not immediately dominate the planet. Between loss and opportunity lies the true mystery, a biosphere surviving the consequences of its own invention while remaking the conditions of survival itself. The mystery of the Great Oxidation does not end when oxygen becomes permanent. Its deepest legacy is the fact that every later ecosystem inherits a planet already transformed by microbial waste. The iron in ancient rocks, the sulfate in the sea, the ozone high above the surface, and the energy released inside living cells all carry the mark of that transition. Long before animals existed, microscopic organisms altered the atmosphere so profoundly that nearly every complex form of life would eventually depend on the consequences. Modern respiration is one of those consequences. In human cells, oxygen travels from the lungs into the blood, crosses into tissues, and reaches mitochondria, where it accepts electrons at the end of a chain of reactions. The process releases energy stored in food and converts it into ATP. Each breath, therefore, participates in a metabolism whose roots reach back beyond the Great Oxidation, to microbes experimenting with oxygen in tiny Archean oases. The machinery has been modified, inherited, and enclosed within eukaryotic cells, but the fundamental chemistry remains ancient. The atmosphere that supports this respiration is not a passive background. It is maintained by living and geological cycles that continue to compete. Photosynthetic organisms release oxygen. Respiration, decay, combustion, weathering, and reactions with reduced material consume it. Over shorter human timescales, the atmospheric reservoir seems immense and stable. Across geological time, it is neither. Its concentration has changed repeatedly, and the modern level is the temporary outcome of sources and sinks that have never stopped moving. Plants now provide much of the imagery associated with oxygen, yet cyanobacteria and marine algae remain central to global production. The descendants of ancient photosynthesizers still occupy oceans, lakes, soils, and microbial mats. Chloroplasts inside plants and algae descend from cyanobacteria that entered another cell through endosymbiosis. The green leaf and the microscopic cell in seawater are therefore connected to the same innovation that once contaminated an anaerobic planet. Anaerobic life also remains everywhere the modern world allows oxygen to disappear. It survives in sediments, wetlands, digestive systems, aquifers, hydrothermal environments, and deep rock. Some of these organisms produce methane, others reduce sulfate, ferment organic matter, or cycle iron. Their metabolisms are not failed relics waiting to be replaced. They remain essential components of planetary chemistry. The oxygenated biosphere still depends upon processes that evolved before oxygen ruled the atmosphere. This coexistence makes the modern Earth a layered archive. At the surface, oxygen supports forests, active animals, fire, and rapid decomposition. A few millimeters below waterlogged mud, oxygen can vanish and older pathways take over. In the ocean, oxygenated water may overlie sediments where sulfate reduction and methanogenesis dominate. Inside the human body, oxygen-rich blood circulates beside microbial communities living under strongly anaerobic conditions. The ancient and modern worlds are not separated cleanly in time. They are nested inside one another. The Great Oxidation also changed the planet's relationship with sunlight. Ozone formed from oxygen absorbs much of the sun's harmful ultraviolet radiation, allowing surface environments to become more hospitable. Without that shield, life on land would face far greater molecular damage. Yet ozone itself depends on atmospheric chemistry and can be destroyed by particular compounds. The protective sky is a product of balance, not an indestructible gift. Fire is another inheritance. Combustion requires fuel, heat, and an oxidant. For much of Earth's history, atmospheric oxygen was too low to support widespread wildfire. Only after later oxygen rises did flame become a major ecological and geological force. Fire reshaped vegetation, nutrient cycles, and eventually human technology. The same gas that threatened anaerobic cells became the agent of cooking, metallurgy, industry, and destruction. Its reactivity remained unchanged, only the systems controlling it became more elaborate. The event also affects how scientists search for life beyond Earth. Oxygen in an exoplanet atmosphere could indicate photosynthesis, but it is not an automatic proof of biology. Water loss, ultraviolet radiation, and other non-biological processes can also produce oxygen. Conversely, a living planet may remain oxygen-poor for billions of years if geological and biological sinks consume the gas as quickly as it forms. Early Earth demonstrates that life can be widespread while leaving little atmospheric oxygen, and that a strong oxygen signal may appear only after long delays and threshold changes. Models of planets around red dwarf stars explore whether photochemistry, ozone formation, volcanic gases, and ocean processes might favor or prevent an oxidation transition. Such studies use Earth's history as a guide while acknowledging that another planet may follow a different path. The Great Oxidation is therefore more than an episode in terrestrial geology. It is a warning against assuming that a planet's atmosphere reveals its biology simply or immediately. Even on Earth, scientists still cannot state exactly how much oxygen filled the air at every stage of the event. Estimates span wide ranges. The order of particular pulses continues to be refined. The roles of phosphorus, methane, continental growth, organic burial, volcanic gases, and climate remain active subjects of research. New isotope systems and better dating may resolve some disagreements while revealing others. The event becomes more complicated as evidence improves, not less. That complexity does not weaken the central story. It replaces a dramatic but unsupported claim of near-total extinction with something broader and more consequential. The Great Oxidation dismantled the environmental stability on which much anaerobic surface life depended. It altered habitats, climates, nutrients, oceans, and atmospheric chemistry. Some organisms lost territory or disappeared locally. Others survived in refuges. Still others gained access to a powerful new metabolism. The biosphere was not simply reduced. It was rebuilt around a chemical division that still structures life today. The possibility that total biological productivity sometimes increased during the upheaval makes the legacy even more revealing. A crisis for one ecological order can coincide with expansion in another. Geological success does not mean universal prosperity, and survival does not mean continuity. What changed most decisively was not the presence of life, but which metabolisms controlled the accessible surface world. The world after the transition remained unfinished. Oxygen stayed low for immense spans. Deep oceans resisted oxidation. Complex cells emerged slowly, and animals appeared much later. Yet the first irreversible step had been taken. Earth could never return completely to the old Archean equilibrium because oxygen production, oxidative weathering, ozone chemistry, and aerobic metabolism had become woven into the surface system. The atmosphere had turned against one ancient order while creating the conditions for another. That reversal is the lasting meaning of the crisis. Life changed the planet without intention, endured the consequences without foresight, and transformed danger into energy through evolution. Every modern breath carries the result of that long negotiation between cells, oceans, rock, and sky. It is easy to imagine deep time as a sequence of finished worlds, each one vanishing before the next begins. But the first oxygen crisis leaves no clean boundary. Its survivors remain beneath mud, inside stone, in dark water, and within living bodies. Its chemistry moves through blood and leaf, flame and ocean, rust and cloud. The lost world is not entirely lost. It is folded into the present. A breath seems weightless. It enters, leaves, and is forgotten. Yet the oxygen within it began as water split by light in cells too small to see. Countless molecules were consumed by iron, sulfur, volcanic gases, and other organisms before any could remain in the sky. Continents changed color. Methane faded. Ice crossed ancient landscapes. Ecosystems narrowed, adapted, and opened again. Over spans beyond ordinary imagination, a poison became a necessity. Nothing in that transformation was guaranteed. Had the balance of nutrients, burial, volcanism, and weathering differed, oxygen might have remained a local trace for far longer. Had life failed to manage its reactivity, the new energy would have remained inaccessible. The atmosphere surrounding Earth is therefore not merely where life resides. It is one of life's oldest constructions, built without design and maintained without certainty. Far below the bright surface, anaerobic organisms continue their quiet work in conditions that recall the planet before oxygen. Above them, forests and oceans exchange gases with the air. Between those realms, boundaries shift with every season and every change in climate. The ancient argument between oxygen production and oxygen consumption has never ended. For a moment, the air around the listener becomes part of that immense continuity, borrowed from a world still changing. And so the final image is not of a world destroyed, nor of a world suddenly saved. It is of a dark shoreline beneath a faint sun, where a thin microbial film releases bubbles into iron-rich water. Most vanish almost immediately. One reacts with stone, another feeds a neighbouring cell, Another survives a little longer and drifts upward. Across millions of years, those small departures accumulate until the sky itself becomes different. The organisms responsible never see what they have done. They leave no voices, no monuments, and almost no bodies. Their memory is carried instead in red rock, in the chemistry of the sea, and in every living creature that draws oxygen inward. The breath passes quietly, but behind it stands the oldest planetary revolution life ever made.